any further, thank you so much, everyone, for coming. Happy to have you here. Happy to be here with my colleagues, Xiaobing and Genya. We'll be talking today about Rapid Novor, obviously, elevating human health by decoding immunity. So we'll do a quick round of introductions here, set the stage, and discuss some of our ongoing work in the antibody sequencing space, particularly directed towards drug discovery, in particular with antibodies. So a couple of introductions and a brief history on us. My name is Anthony. I'm the Director of Business Development here at Rapid Novor, and I represent a lot of the business side of the equation here at Rapid. And I'm also joined by my colleague Xiaobing. Xiaobing, can you say hi?
Hi, everyone. Thanks for joining us. And my name is Xiaobing, and leading the antibody discovery effort in Rapid Novor.
Very good. Thank you so much. So we'll be hearing from both of us today, and let's do a quick round of a history here at Rapid Novor. So for those of you who are unfamiliar with us, Rapid Novor has been in the business of antibody sequencing using mass spectrometry and really a proteomics-based approach now for about 8 years. So we were founded in 2015, where we were really starting to just offer the monoclonal antibody sequencing service, REmAb. But obviously, over the past several years, we've been able to innovate and improve the fidelity and throughput of the technology by delivering such advances as the wild technology for discriminating between isoleucine and leucine, which is obviously pretty critical when trying to determine the precise amino acid sequence of a given antibody and protein. Then, moving forward, we were able to actually apply this in a clinical setting towards our EZM multiple myeloma assay, where we can derive the monoclonal antibody secreted by myeloma patients directly from their serum without having to do any kind of cellular-based sequencing. More recently, you're able to sequence, many thousands of antibodies to date, but more importantly, launch our REpAb technology, which we'll be speaking on in a bit further, and then subsequently unveiling our protein-only polyclonal antibody sequencing technology, as well as our EZM paper in the clinical setting. So to date, in 2023, we're well over 70 employees. We've got many teams now based out of our Kitchener office. That's just a little bit outside of Toronto here in Canada. We've got well over 20,000 square feet of lab space and over 15 mass specs now humming along in the lab. Our track record, I think, speaks for itself as well. Hopefully many of you are clients that are joining us. If you're not, suffice to say that we've been able to work successfully now with many companies, including most of the major top pharmaceutical companies globally, many academic institutions such as MIT, Harvard, Oxford University, UCSF. I don't need to list them all, but I think you get the point. We've been able to sequence not only monoclonal antibodies, as I said, but certainly polyclonal antibodies as well. We were the first team to successfully complete such a workflow and demonstrate such success by recombinantly expressing those monoclonal antibodies that we found and showing their performance as compared to the polyclonals, which we do have some data to show a little bit later on today as well. This has really been not just the fruits of 7 or 8 years worth of innovation, but really 20 years plus. Our co-founders have been in the business of mass spec-based proteomics sequencing and early software development for well over 20 years, and that's really helped us to serve many different clients and secure some meaningful intellectual property as well. So in terms of what this has yielded, in terms of our service offerings, we have what we would describe as our core services, the next generation protein sequencing based services, as I mentioned, REmAb for sequencing a monoclonal antibody of interest, as well as REpAb, which is more of an antibody discovery based approach, but certainly could also be used in sequencing just a polyclonal antibody that you may already have access to internally. But just getting an antibody isn't the end of the story. Oftentimes, oftentimes we also need to characterize those antibodies as well, which is why we've been able to expand our service offering, mostly on the mass spec front, but indeed in other ways as well, by offering our other services, which are MatchMab, which is more of a sequence confirmation, like a quicker version of our REmAb service, where we try to run our mass spec analysis against antibody A and antibody B, and we are able to determine, if these are identical sequences or not, without having to perform a multi-enzyme digest. This is it's a bit of a simpler workflow. HDX mass spec for epitope mapping. I'll speak a little bit more about this a little bit later on, but in short, it's a service where we are able to discretely determine the binding amino acid residues between two molecules. It could be a protein, it could be a DNA and a protein of interest. It could be a small molecule. These are all reasonable cases for the HDX-MS workflow. We were able to work with SPR as well for determining the kinetics of two proteins or two molecules of interest. We've also been able to unveil our cloud-based proteomics software, Novor Cloud, so that if you have mass spec data, you too can run some of our analyses on the cloud. And lastly, our glycan analysis, which is rolling out a little bit later on this fall, where we can in fact take a look at the glycan present on your antibody of interest, and in some cases also your proteins of interest as well. Feel free to ask some questions about that a little bit later on. But with that in mind, let us go a bit further into the next generation protein sequencing approach. So, we really view this as a way of like shifting R&D. I don't think that we're in the business to say that this is the only way forward. I think it's fair to say that this is going to be a complementary approach. But I would certainly say that we view this as linking into, let's say, a fashionable trend in antibody discovery, which is that we're starting with function. Being function forward, I think, is always of interest. But I would say that luckily we're a little bit different. Whereas a lot of people end up screening for function using cells of interest, we're obviously screening for function using the antibodies themselves, the proteins themselves. And we would certainly argue that there are advantages to doing it in that way. But at the end of the day, I think we're really just trying to say that starting with the antibodies that you really want can certainly ease some of the pain that some people experience in the antibody discovery process. So how are we doing this? Well, our core technology is really based off of this premise, where we do work with a purified monoclonal or polyclonal antibody. We do a multi-enzyme digest, which is to say that we take your intact proteins of interest, we cleave them into many shorter peptides using many different enzymes, which selectively cleave at different amino acid locations. And once we've generated these peptides, we fly them on our LC-MS instrumentation. And once we have done this, we will generate many spectra, thousands and thousands of spectra, which are then analyzed by our Novor algorithm in order to do the peptide sequencing. And once we have a sufficient number of highly accurate peptides, we can actually piece them together into a full-length contiguous protein sequence. Canonically, it's going to be a heavy and a light chain, at the end of which we will put these sequences into a report and send them to you for expression, engineering, what have you. So in a monoclonal antibody workflow, looks something like this, where again, we're starting off with your antibodies of interest. We always run a gel first because it is wise to do such a thing. We always want to make sure that we're really starting off with a monoclonal antibody, or if there are any surprises, like perhaps an additional light chain or a glycosylated chain, this is of interest to us. So indeed, we run the gel first to kind of observe what is really present in the sample. But suffice to say that if it's a monoclonal antibody in the true sense, we will be able to sequence it using only about 100 micrograms of monoclonal antibody, perhaps less, depending on the situation. Purity being one of them, the main factors that we're considering in this. But yes, surely just to say that at the end of such a workflow, you'll receive the antibody sequence report, which will give you the precise amino acid sequence of the heavy and light chains, as well as the paired heavy and light chains. This should be relevant to your project. And moving on from that, of course, as I said earlier, we were able to really work in a more complicated situation, which is the polyclonal antibodies. So, again, we really view this as a complementary approach to the existing modalities. We would argue that, starting off with the protein that you like to work with could be another way to go about this problem of finding a good drug like monoclonal antibody against your target of interest, or just a good binder in the case of like a diagnostic or a research reagent. But of course, we've been able to work with this workflow now over the past several years. We've been able to apply it against, luckily, many different species. This isn't just a cute diagram. We actually have sequenced antibodies from each of these animals. So canines, goats, alpacas, chickens, sheep, rabbits, humans. I feel like I'm missing one or two, but suffice to say that we really have no limitations with respect to the species of interest. Projects get a bit harder, let's say, but nothing that we haven't been able to already work with in the past with respect to like different species. We're fundamentally species agnostic. In short, the reason is because luckily antibodies are made of amino acids, which are a fundamentally universal building block, right? So when we actually run them on the mass spec, we're able to always do the peptide sequencing and the assembly of these residues. Whereas in a cellular-based approach, you need to develop primers, you need to test them in a very specific fashion for the molecules of interest, like the B cells from some species of interest. So we don't really have this limitation on the mass spec side, although it is always helpful to work with species for which there are means to do the cellular-based sequencing if that is of interest as well. So of course we're saying that this method is complementary to other existing modalities, like your typical hybridoma-based approach, whether you're using a display approach with phage or yeast or single B cell sequencing, all can yield hits. Of course, we wouldn't disagree with such a statement. In fact, they've been proven out quite a bit more, I say, than our technology. But what we will also add is that our technology is delivering something quite unique, quite differentiated, and we have some data to I think show that a little bit later on as well. So let's say that there's a new compartment, a new way to access the immune repertoire using the proteomics approach that you cannot obtain using a cellular-based approach, at least in our experience. So the REPAb workflow looks a little bit something like this, kind of a typical antibody discovery workflow, if you will. You do the immunizations, you test your antibody for titers, but of course in our workflow, one thing that's a little bit different is that we're always going to be purifying the serum against a target of interest, or targets of interest, I should say, depending on the situation. Once we have this antigen affinity purified, preferably IgG polyclonal antibody, we can then put it through our polyclonal antibody sequencing protocol using the mass spec based approach. And then subsequently, once we have a list of not only heavy and light chains, but also paired heavy and light chains, we just have a list of IgG sequences. Once we have that list, of course, we can then do the monoclonal antibody recombinant expression, perhaps with some of our partners we like to work a lot with. And then once we have those monoclonal antibodies in hand, you can then do the profiling and characterization, which could be like the SPR, the HDX workflow, or perhaps like the constellation analysis, as we alluded to earlier. With a bit more detail, this is what we were showing earlier, where we do the multi-enzyme digest, we do the mass spec sequencing work, and then we yield the full-length monoclonal antibody candidates, whether that's just from the protein-only approach or whether you optionally decide to send some B-cell NGS data as well. Okay, so the idea is really to take these these ideas, these potential sources of valuable antibody targets, and then really turn them into drug-like molecules. So fundamentally, as I said earlier, REPAb is species agnostic with many successful projects across many different species. You're really, in our opinion, getting more shots on goal by accessing the complete functionally expressed antibody repertoire in a cohort, and we'll be able to show why we would make such an argument in a few slides. And it's another way to access the only truly human antibody repertoire working with serum samples from patients. Of course, the cellular-based approach could similarly work in this way, but I think, again, the difference is in the functionally expressed antibodies that we are observing using the mass spec-based approach as compared to other approaches. And what I mean I think what we mean when we say truly human is that, of course, there are transgenic animals. Of course, you could work with mice as well, but they don't really capture the entire breadth of the genetic library in the same way that an actual human individual does, because they always work with a subset of the human genes that are spliced into their antibody repertoires. And we also believe that this is a way to select the most robust monoclonal antibodies, as they are secreted in serum and shipped to us, and they are still there at present. I think there's an argument to be made about the robustness of a protein analyte versus the cell themselves, and we'll get into that a little bit later on as well. So lastly, I won't say this anymore too much. The complementary approach is really the notion that we're proposing here, and we will have I think a fairly cohesive way to present this as well. I really view the polyclonal antibody sequencing technology as a way to kind of overlay or work with those other existing technologies in a truly harmonious kind of fashion. And of course, as I said earlier, protein really is a robust analyte. I think many people who have worked with cellular-based approaches can comment on this further. B cells are tough to keep alive. I think if you're using a platform that does the single cell screening and sequencing, it can be challenging to keep those cells on the chip of interest and really screen them for all the functions that you would like to, without losing a huge portion of those cells. Whereas the protein itself, as we said earlier, it's really robust. We can sequence an antibody that's been shipped to us from Australia. That's about as far across the planet as you can go. We've been able to work with cryopreserved samples that have been preserved in a freezer for decades. We've really been able to work with samples that are put through a rigorous, let's call it screening or stability test, if you will. So I think that in this way we've been able to demonstrate that the protein is indeed a robust analyte, and you can work with it in a plethora of screens up front before selecting for the final functional activity that you're really keen on. And as I said earlier, we've been able to work with these cryopreserved samples, not only the fresh immunizations. So if there are, indeed good antibody reagents that you've been able to work with or source internally through your own immunization campaigns, they could easily be candidates for such a project with us. Although I think that we can comment a little bit further on what is a good or a bad antibody to send us offline. But indeed, it is the case that we've been able to work with such projects in the past. And so this is where I start to turn things over to Xiaobing.
Thank you. Yeah. So in the next section, I would like to further introduce Novor's antibody de novo sequencing technology and show you a few examples of the power of protein sequencing in antibody discovery, including the expansion of your candidate pool by proteomic discovery and providing the proteomic evidence to aid your candidate selection, more like a focusing. When using the mass spectrometry to do the proteomic analysis of a serum proteins, the typical strategy is to construct a reference database from the genome sequence. But this strategy is not useful for interpreting the antibody-derived mass spectra. Why? It's because there's a high antibody repertoire diversity exists. For example, the heavy chain CDR3 hypervariable region was encoded by the VDJ gene segment, and the diversity was caused not only by the recombination of different gene segments, but also introduced by introducing the P and N nucleotides at the joint. We called it junctional diversity. So the non-templated nucleotides were added by the TDT enzyme, giving rise to the random number of amino acids not coded in the genome in the germline sequence. so the genome sequence cannot be used as a reference. Another approach is to use the B-cell mRNA sequence to build the reference database, which will reflect the transcriptional diversity. However, existing mechanism for antibody diversification can yield an astronomical number of possible sequences. In theory, there's over 10 to the power 13 in human. And this number is a few orders of magnitude more than the total number of B cells in the human body. So the mRNA reference database still cannot represent the full diversity of antibody repertoire. The use of a de novo peptide sequencing for mass spectral interpretation does not require a reference database, therefore offering a promising solution to this problem. We address this challenge with complementary enzyme digestion, multiple tandem MS-RAN, and a different type of MS-MS fragmentation to yield rich sequence information from the tandem spectra of the peptides. Here is an example of a CDR3 sequence that was successfully de novo sequenced from the tandem MS-PRM spectra. So the de novo approach has allowed us to resolve the sequence diversity during this antibody discovery exercise. Next slide. In our protocol, the antibody was digested by multiple complementary enzymes, and the tandem mass spectra were produced by high mass accuracy mass spectrometer. With the aid of our proprietary software, full and accurate sequence of the heavy and the light chain can be reconstructed with the tandem spectra of the digested peptides. So the use of high-resolution MS and the powerful machine learning software provide large benefit in sequence coverage and accuracy in our workflow. Our protocol yield full and deep sequence coverage, as demonstrated by this coverage map, in which you can see the top line indicating the amino acid numbers, and that the green color indicate the variable region, the framework region, and the orange color are 3 CDRs. The blue color is the constant region. So the peptides were digested by multiple enzymes that show in different colors, and the overlapping peptide provide 100% coverage of the full antibody sequence. And finally, our machine learning algorithm provide high confidence, heavy and light chain pairing information as well. Yeah, next slide. So as we mentioned, the extraordinary number of antibody repertoire diversity posed a major challenge to our practice in antibody discovery. Actually, the B-cell heterogeneity add another dimension of the complexity to this finding a needle in a haystack exercise. B-cell development involved a series of stages which changed the location. The life stage began in the primary lymphoid tissue such as bone marrow, and the maturation in the secondary lymphoid tissue. The endpoint of the B cell is either memory cells or antibody-producing plasma cell, and those cells traveled back to the bone marrow, spleen, lymph nodes, and the mucosa-associated lymphoid tissues. So you can see the peripheral blood B cell compartment only represent a crossroad in the B cell trafficking. And the circulating B cell only represent 2 to 3% of the total B cell population in humans. And the bias can be easily introduced using peripheral B cells to study the whole antibody repertoire. Instead, sequencing antibody at the protein level directly reflects the circulating antibody repertoire and the relevant abundance in the specific conditions. So proteomic profiling expanded the antibody repertoire heterogeneity and enable the endogenous humoral antibody discovery. In the next slides, we will show you an example. In one of our projects, we studied antibody sequences discovered from de novo protein sequencing and the B-cell sequencing from either splenocyte or the PBMCs. And we found, well, sequence agreement exists between the different discovery approaches. There are significant number of protein sequences that could not be found by the B-cell sampling. Some proteomic discovered antibody sequence represent the unique clusters of antibodies demonstrated by the phylogenetic analysis on your right-hand side. Those results convince us that the combination of different antibody discovery approaches will largely enhance the antibody discovery efficiency and improve the success rate. So in the next slides, we said combining the genomic and the proteomic approach will enhance the success rate, but also will not only expand your candidate pool and also can facilitate the identification of biological relevant antibodies. In the past decade, especially during the COVID-19 pandemic, remarkable breakthrough had enabled the discovery of antigen-specific monoclonal antibody directly from human donors by mining the human B-cell receptor repertoire. Sequence analysis of a BCR repertoire is a rapidly evolving field. Various computational approaches have been used to identify the antigen-specific antibodies from a very large repertoire sequence database. However, as we discussed before, there's a functional disconnection between those assay-screened antibodies present in the serum and antibody-producing B cells present in difficult to reach compartments. So our approach can address this issue by overlaying proteomic evidence with NGS read to focus on the biological relevant candidate in the discovery workflow. When studying the antibody repertoire in a hyperimmunized animal, as we show in the next slides, usually we can see clear evidence for clonal expansion, hypermutation patterns, sequence cluster, and immune conversion, etc., etc. But the antibody response is much more complex in human due to the heterogeneous population of human subjects exposed to multitude of antigens in an extended period of lifetime. For example, when we look at the germline usage of a proteomic discovered antigen-specific antibody sequence in naturally exposed human and in antigen hyperimmunized animal, the first 50 most abundant clones were analyzed, and we can see restricted VH germline usage in animals, but with only limited evidence can be found in human. And there's clear inter-individual variations in the VH usage in different human subjects. So in some of the human projects, when our partner or collaborator were facing over 10 gigabytes of NGS read, our proteomic profiling could provide tremendous complementary evidence for candidate selection and enable the antibody discovery directly from human serum. So due to the high sensitivity of the instrumentation, the mass spec-based proteomic antibody discovery require high quality and affinity purified samples. Actually, the sample preparation and the affinity purification is also a functional selection step for our proteomic sequencing workflow. By incorporating carefully designed enrichment strategy, a subset of antibody can be selected and enriched with very specific functions, and which will greatly increase the success rate for finding the right candidate. For example, in this ADA control developing project, the animal were immunized with a monoclonal antibody, and the immune serum were purified by a standard proteage method. The polyclonal antibody underwent a negative selection using an antibody with the same constant region as the immunogen, but a very different variable region. And a positive selection was followed using the original immunogen for enrichment. After the protein sequencing, when we identified the 30 most abundant clones and expressed 18 clones in the first batch, and in the next slides, you can see we found 16 clones out of this 18 bind to the original immunogen, but none of the clone bind to the antibody with the same constant region. So in the next, So in this study, we used a very simple workflow and discovered a rich pool of candidates with selected function of anti-idiotypic features. And I will pass back to Anthony, give you another case study example.
Great. Thank you so much, Xiaobing. Appreciate it. Yes, so as Xiaobing has said, we have one more case study to go into, but I'll quickly preface that by getting into a bit more detail about the nature of the characterization. So the nature of it is actually based off of some workflows that we've done internally. So one of them being HDX MS epitope mapping. For those of you who are unfamiliar, this is a great suite of technology for finding the binding amino acid residues between, like so canonically, like an antibody and an antigen of interest. The nice thing about it is that it can work with linear epitopes, conformationally specific epitopes, as well as structural epitopes. Has extremely high resolution, and it is relatively quick. It can be used for a number of applications. I think that, like I said, the typical use case is the antibody and the antigen of interest, usually for like some kind of patent filing, but it could certainly guide some kind of selection of an antibody against the target of interest. In short, the way it works is we label an antibody, an antigen complex of interest with deuterium, and then once we have it labeled with deuterium, the binding amino acid residues will be obfuscated or occluded from the deuterium. And then once we are able to actually digest it using some enzymes, you can then find some peptides which will not be labeled as such, and then we can infer the binding amino acid residues in this way. So again, it is a highly accurate and highly high resolution method down to individual amino acids in some cases as well. So and the important characteristic as well here is that it is we're looking at the molecules as they interact together. It's not an individual case where we're removing one and then the other, or removing an individual amino acid. This is your molecule against the antigen of interest. Okay. And in addition, we've also been able to characterize some of our antibodies using SPR. I think most of us are fairly familiar with the notion of SPR. Obviously, it's real-time monitoring, it's label-free, doesn't take much material, and we can use some fairly complex samples as well, like an antibody and some other analyte of interest. So we're going to immobilize our antibodies or our analytes against the chip, and we're going to flow over either the antibody or the analyte depending on the situation. And in this way, we can infer the binding kinetics from the readout of the device itself. We personally work with the Nicoya Open SPR system. They are a close neighbor to us, located here as in Kitchener, Ontario as well. But indeed, the results are, I think, fairly consistent with what we would observe in other types of machines as well. So again, we applied this kind of technology against a particular case. So in this situation, we actually used it against camelids. We used it against in an alpaca antibody discovery project, much like some of the other products that we've been able to observe. We did the antigen purification against our targets of interest, the mass spec-based sequencing, and then we read out the full antibody sequences. One nice thing about working with camelids is that usually we're just looking at the heavy chain antibodies. And I think that we're quite fortunate as well to be working with the protein in this case. For those of you who don't know, camelids actually produce a number of different isotypes with respect to their heavy chain antibodies. And so we're actually able to segregate different heavy chain only antibodies based off of the isotype of interest, which can be interesting as it relates to the hinge region. Some camelids produce a short hinge, a regular length hinge, and some of them have in fact even a longer hinge, which can have some interesting effects. But I think that that's a discussion for a different day. So in here we are going to show that the serum was collected from this animal that we immunized, and the immunogen in this case was actually a rabbit polyclonal antigen. Then we de novo sequence these antibodies. So again, we did characterize them using the SPR. We actually had obtained 10 different sequences from this project, and from that pool, 6 of these MAB IgGs were selected for synthesis and further characterization. So here we can see that on an ELISA, we were able to confirm binding for many of these antibodies against our target antigen P17 as well as a CD3 epsilon. So in this, we had about 3 that were strong binders against the P17. That is our R4, R7, R9, and a couple of them that also bound against CD3. So we're looking at R4 again, which is nicely cross-reactive, and then R9 as well. And so we started to kind of pool them on where they were binding, but the SPR, we hope, tells a more complete tale. So again, we ran these 6 antibodies, which displayed a range of affinities towards our antigen of interest, our P17. And then we could really start to discriminate between some of the top binders, which had high stability and high affinity, as well as others that had the high stability component, but the kind of average affinity. This is our R4, which is nicely cross-reactive. And similarly for CD3 epsilon, we noticed that R9, again, doing really well against both of these in terms of their binding affinity, which was useful to us in terms of further characterizing, then using a iso affinity plot. So in this way, we're not just looking at the on rate or off rate individually. We're really using both and creating a ratio to give you the final KD value, and then plotting them against this iso affinity plot. So we can see that R4 and R9 are really the higher affinity binders in these instances, and also that R7 is more selective towards the P17 monoclonal antibody. From there, we did do a bit of an epitope binning approach. So we did the immobilization of our P17 antigen, and then we saturated them with one of the antibodies of interest as we moved our way through. So we in fact ran 16 actual reactions. Obviously, there's the 3 different reactions, but we always actually run a reaction here just to confirm the result that we expect to observe. So in this way, we can actually observe that some of these were strong binders, and some of these were some of these were strong binders in that like R4 when saturated against the antigen of interest. continued to bind against the antigen of interest in the presence of R1, and similarly R4 continued to bind against the antigen of interest in the presence of R7. But it's interesting to observe that when R7 was saturated against the antigen of interest and R4 was flowed over as the analyte, it actually didn't really push off, or sorry, it did push off the R7 antibody in this way. So we see that it is interesting to not see the inverse. Sorry to see the inverse relationship here, but not see the same blocking interaction here. And we actually believe this may be due to a kind of steric hindrance kind of mechanism in this particular instance. And then we were able to use this this binning data and create a nice graphic here as well to demonstrate the nodes for which the antibodies are binding in tandem. And then lastly, we were able to actually overlay some of these expected results from the binning experiment against the HDX data as well. So as I said earlier, we were able to determine the binding amino acid residues from HDX, and in fact, we did that here. So you can see the R7 binding residues are highlighted in the green, and similarly, the R4 residues are highlighted in orange. So we interestingly observed that the R4 binding residues are there are some on the FC, but indeed there are some as well that are focused on just here under this this cleft adjacent to the R7. So we think that this might have something to do with the reason we're seeing the binning results that we had observed as well. And we've actually gone a little bit further and created a biparatopic molecule, but we just have yet to test it at this moment. We're just in the process of that at this moment still. So we look forward to sharing the results of that as well in future commentary. Okay, quick summary here, and then we'll be open to take some questions from you guys. So obviously there are a plethora of applications for which antibody discovery can be applied more broadly, but we've actually been able to, I guess, see a lot of interest, in particular in the immunoassays, the IVD market, interestingly as well in the veterinary medicine space as well. I think that being species agnostic and being able to work with things like samples from canines and other kinds of farm animals, so not just your companion animals, but also your more farm-based animals as well, have been able to serve us well in terms of addressing that kind of a market. But we've certainly been able to work with a handful of clients in the immuno-oncology space as well, and doing truly human-based antibody discovery and other antibody discovery projects more directed towards therapeutic applications. In terms of how we typically operate, we like to do a lot of discovery partnerships, but indeed we do work, I mean, in principle in two different ways. We can do cash only kind of fee for service arrangements, no royalties, like no milestones, none of that. Is certainly like a a typical kind of option that people choose to work with us. On the flip side, we do also make such options available in a more kind of collaborative scheme where we are working together towards developing a drug of interest, and we would share in that risk, and then hopefully work with another partner to do the discovery stage partnership thereafter as well. So we're not married to one specific way of doing business in this sense. And then of course includes our ability to do the antigen design and coming from ideation through the sequencing work and actually delivering some hits. We leverage our purification technique expertise in these ways, and then further characterization using HDX or maybe the glycan workflow, or some of our other characterization services as well. Whatever needs to be done towards the goal of ultimately bringing a drug like molecule to IND. And that is it. This is my contact information as well as Xiaobing's. So feel free to reach out to either of us. We'd be happy to hear from you. And I think now we can open up the Q&A for discussion. So I will look at some of the questions here. I see them. And Genya, I don't know if you want to chime in here as well.
Sure. So yeah, our Q&A is open. So if you have any questions, feel free to drop them down and we will get to as many as we can. We do already have a few in queue. So our first question today is, how much antigen enriched material would you need? And how much serum would that typically correspond to?
Sorry, I'm just trying to see. So is sorry, just so I'm reading the question while you read it. Is this the question from Moritz?
Yes.
Okay, right. Okay. Yes. So how much material would you need? Well, we typically ask for about a mg of antigen affinity purified material. Now, I can hear the reaction. So there's a lot of material, right? So I would say that it really depends on the specific project as well. That's our ask, is like 1 mg of antigen affinity purified IgGs. We have in some instances worked with a bit less, but I'll say that we really need about a good portion of that material towards the goal of sequencing, and quite a lot of the material is also used for the specific purpose of chain pairing experiments. So we use obviously proteomics to do a lot of our work, and in fact, on the chain pairing side, similarly, we use a lot of material towards that goal. So that's the ask. We can certainly have a more extended discussion about what are the edges of that kind of range offline. So I be happy to follow up with you afterwards, Moritz. I would also say that how many functional sequences do I expect to get? That is a loaded question that is highly variable. So it really varies for a lot of reasons. I don't mean to kind of escape the question, but I would just say that in some instances, some of our projects have yielded a few antibodies, and that could be because there's a very convergent antibody response after repeated immunizations in like an animal of interest. But as Xiaobing alluded to earlier, human patients are much more complicated. And even some of our animal immunization projects, even with a small antigen, have yielded like dozens of hits, like 40, 50 hits. So it really depends on the biology of interest from like an immunization perspective and also from a host's perspective. So again, happy to have an extended discussion with you offline about that as well. But we typically do request that they come from a single donor, as you framed in your question, and the process usually takes a few months, so 2 to 3 months to do the, if you sent us serum to you receiving sequences in your hand, usually about 2 to 3 months to do that. Obviously need a few more months to do the expression work as well, unless you can do it in-house. It might take a little bit less, but that's in general the timeline that we're discussing. Okay, I guess I can work down the list here. So I've got a question here from Rafal. Does the profiling and characterization step give antibody glycosylation profile? I would say that it gives you like a rough outline. Like we're not going to be doing the sequencing of the glycan. Like we wouldn't be able to specifically tell you like all the different sugars in order and in the structure of it per se, as much as we can work with known structures like your typical G0, G1, G2, and these kinds of variants that we would expect to see out of a typical monoclonal antibody expression. Okay, question from Yimin. How do you know your antibodies reconstructed resembled from the mass spec results represent the naturally occurred antibody? I would say that is well, I don't know, Xiaobing, you want to try this one?
Sure. I think first of all, we are using naturally occurring antibody as our starting material. We are instead of finding a B cell, like we use this antibodies present in your polyclonal mixture, and you already tested this mixture before sending to us for the specific function and the titers, etc. Another thing is I think probably you're also asking like the because there's small peptides for the mass spec, so then we construct them together. But as you can see from a webinar, the peptide was digested by a lot of enzyme with a different cleave site. So the overall coverage, because it's overlapping coverage, is much higher. So we, using our proprietary software, we're able to accurately reassemble them together. Yes, here. Yeah, so it's not only like trypsin digest, like it's all the same length, but all the peptides different length. And in another level, the mass spec technology, like I'm not a mass spec expert, but we have a lot of mass spec scientists. So it's multiple mass spec run in a different instrument, and it's not only bottom-up approach can be used, and also other middle down, like top-down approach. So there's involved a lot of experiment, a lot of mass specs run here. So it's we present you a coverage map. It looks very simple, but behind this coverage map, there's a lot of mass spec to run. So that's why, like, we can provide, we can be very confident on the sequence coverage, and also know, like, we assemble them together is the correct, accurate sequence.
Yeah, for sure. So let's move on. And anonymous attendee asking how the thermo-astral Orbitrap equipment will generate, well, over 100 gigabytes per hour worth of data, might impact our processing speed and efficiency. I would say that we would have to purchase this instrument in order for it to, for us to really have an opinion about it. I think that our current workflow is fairly robust and works well, but I mean, of course, we we like to buy the new toys from Thermo as well. So perhaps when we have a chance to use that instrument, we can comment a bit further on it. Okay. Deshu, the price, yeah, we can always talk about price offline. I think that it's just a very custom kind of workflow. So I think it's best served to just have a more nuanced discussion about the specifics offline as well. So Vikas is asking, you obtain 100% sequence coverage by digesting with only 5 enzymes. How long does the analysis take from start to finish? I would actually say that we don't just achieve 100% coverage, we achieve 100% accuracy. We actually, in this specific graphic, are demonstrating that we don't just have 100% coverage with, I mean, one peptide, we have hundreds of X coverage across, a large, at least I can't count all these peptides, but I think it's safe to say there were at least like 100X in some of these instances across this sequence that is completely covered by the sequence. But I think that in principle, we always try to achieve at least 30X coverage with individual peptides across every single amino acid. That's really because you need redundancy using proteomics. Perhaps you're familiar already. I don't need to explain this too much, but I would just say that, with peptide-based sequencing, not every read is created equally. Some of these have higher error bars than others, and so you really gain a lot of accuracy by reducing by improving the redundancy and by having more and more coverage, which is why we've really specifically focused on that aspect of this sequencing approach. Over the past 7-8 years, we've developed a number of wet lab protocols to improve coverage, and a lot of bioinformatics tools as well to improve coverage. So indeed, the ability to achieve 100% sequence coverage is always kind of a bare minimum in a lot of experiments, but I'd say from our end, we're not so focused on that as an end goal as much as we are 100% sequence accuracy. And we've been able to demonstrate that by sequencing thousands of monoclonal antibodies and dozens of polyclonals now as well. How long does it take? I'll throw back to you, Xiaobing, after I answer this. How long does it take? It takes about usually a week or two to do a monoclonal project, and then, like we said, a couple of months, 2 to 3 months to do a polyclonal project as well. Xiaobing, you want to add something?
Yeah, I'm just saying, like, in this coverage map, like, we show 5 enzymes, but we use much more than this one. Yeah, it's just the example here is from the 5 enzyme. Yeah, if we put all the enzymes here, we won't be having enough space to show you the coverage map. And how long it take, and that is also, it's very project dependent. And also, like, usually for the polyclonal sequencing, we put a middle checkpoint first to check your sample and to see if your sample have enough that CDR3s can be detected, and then we will proceed with next step.
For sure. Yeah. Thanks for bringing that up, Xiaobing. As she said, we this is a two-stage process. We definitely triage that project up front in order to ensure that it is even sequenceable in our hands. Otherwise, if it isn't, we would stop the project immediately at that point. Okay. Answer live. We did that. Carlos is asking, can we talk a bit more about the deep learning model for the heavy light chain pairings? Unfortunately not. It's a bit of a proprietary trade secret, so we can't really share too many details there. We can certainly share the validation, if you will. I'm showing that it has worked, and I could maybe detail a bit further about what that method looks like offline as well. So we'd be happy to chat about that a bit more through email or through another call of our own. Okay. Moritz, thank you. Jacqueline, how do you couple the heavy and light chains together using the same antibody using the MS data? Again, it's not something that we can comment on too much here, but I would just offer a bit more. I don't like to dodge the question, but because we work with the intact protein, in a lot of cases, we run like a native gel, for example. We can in such an experiment, and we have a handful of experiments that we typically deploy. You can imagine that we work with the intact antibody. We can work with the correctly paired, the cognate paired heavy and light chains. And when we have a number of experiments in which we fractionate antibodies and we can observe them consistently using the mass spec-based approach, we can infer that they are indeed paired. And then the ultimate test for us is always expressing them recombinantly and testing them. I would say that this isn't going to be the same as using a cellular-based approach where you're sequestering an individual cell, and then sequencing that. But I would say that at the end of the day, what you're really looking for is a good antibody. I think that one day we would like to run the comparative experiment against a single-cell sequencing-based approach, and really compare our chain pairing technology. But insofar as the antibody binds to its target, it expresses well, it's drug-like. I don't think that I think that we're serving the goals of the project quite well. So yeah. Answer this. How do you match? Okay, I'm gonna thank you. I missed the overlap nature. No worries. It's all good. Thank you, Yumin. Where does the turnaround time for the de novo sequencing approach? Yeah, I think we've answered this now. About a week or two for the monoclonal case, and then about 2 to 3 months for the polyclonal case is generally the timelines. But of course, if you have to do immunizations first, if we have to do expression on the back end, these things can kind of make the timelines a bit longer as well. But always happy to have that conversation offline. Okay, cool. So that represents the final question that has been posed. Here we are at the top of the hour. I think we're all set. Anything else, Kenya?
That is everything. The webinar has been recorded, so that will be provided to you shortly afterwards. And then you've been given Anthony and Xiaobing's contact info. Maybe we can go to that slide again. So if you have any questions, you feel free to reach out to them. And thank you for joining us today.
Yes, I'm going to skip forward here. There we are.
There we go.
Yeah, of course, you can always reach my phone and reach us by email. Happy to hear from you guys.
Yeah, looking forward to talking to you in the future.
Great. Thank you.
Okay. Bye.